Well, I will be talking about the fundamentals of the applications of the biosensors. Now I talked about the biosensor. So is it that biosensor is going to be useful only for the in what you call the military? No, biosensors are useful for the several. What is biosensor is that it is the instrument which is capable of measuring the analyte of our interest. It may be cholesterol, it may be glucose, it may be simply sodium, potassium or pH or oxygen concentration, whatever may be the. This instrument will be able to replace your normal chemical analysis that is done in the laboratory. If anybody goes to a doctor and says I got a fever etc., sometimes he may give a medicine based on whether why you got fever etc., depending upon the local circumstances. That's how the doctor take a declaration. Of course, they will know that what type of disease coming, what symptoms, everything. But it is not yet always possible to know from the symptoms. When it is not clear, then only I think they will send the patient to a pathology laboratory where they do for the dengue or chikungunya or the malaria or typhoid. They will test it. They will give a report to the doctor, and then the doctor will be able to tell. So this takes a longer time today. Second thing is that the time taken for this testing of these things is going to be quite high today with the existing laboratory analysis, the procedures, and the patient has to go to some other pathology laboratory etc. This happens, but in future, if the biosensor really work, then what happens? A patient will walk into doctor's this consulting room. The doctor may prick his this hand from a drop of blood, and he knows what type of disease he has got it. This is the ultimate goal of this one. It is not only simple disease; there may be so many genetic disease, everything which can be found out with the help of this one, with the help of the biosensors. Now, I will try to introduce you to the what is the biosensors. Biosensor is the one actually basically has it is a conglomeration of different types of the sciences, everything. One of them is called a transducer. It has got different parts. Different parts are put together to call it as a biosensor. So in this one, in biosensor, if anybody is having, the first part is going to have transducer. The transducer is the one which will be able to convert any changes that is taking place around it. It may be a change in the weight, it may be changing some electrochemical property, it may be changing the optical property. That changes will be felt by it, and then it will give an electrical signal which can be manipulated later. So basically transducer is the one which will convert one form of energy into the other form of energy only, and the transducers are normally used as electrochemical because they are cheaper, and then there is a optical, gravimetric and colorimetric, and then so many other new varieties are coming. And this is the subject of different the sciences. It is physics, chemistry, biology, the chemistry, physics, chemistry, electronics, and electrical. But so many people can be contributed in for developing of the transducers. The transducer being used, even though if you are driving a car, you can have that speedometer. All these things are nothing but nothing but the transducers, which will give you the data about the speed, how much consumption of petrol. All these things they give. They are nothing but a type of transducers. Now, in the case of the biological, what happens is that it is necessary to find an analyte in a blood, or maybe in a urine, or it may be maybe in an environmental sample, maybe in some other matrix. One has to find out. For example, somebody wants to find the glucose in the blood, or uric acid in the urine, etc., or in the blood, whatever may be the case. Then he should recognize them. So for recognition, you need a recognition molecule. So what is happening in the sensor? One is a transducer which will be able to convert something to the an electrical signal, but how to know? For knowing it, it need a recognition molecule. These recognition molecules are given in the cup of blue color there. They will be enzyme or antibody or receptor or microorganism or the nucleic acid etc., which can find out. It is not almost all bacteria, everything will be having the made of amino acids and DNA etc. So chemically they are not different, their functionalities are different. So in order to know what is it is necessary to use only biological methods to detect the particular species, and that is called the recognition molecule. And now there is a complex molecule is there, in medium is there. You say this is suppose glucose actually. If you want to find out, you need a specific recognition element. So in this case, it is glucose oxidase. To find out that it is one, it should not give a signal to the others. So there is a definition is there. It only says that you have to keep the it should be integrated device. It should not be in the different parts. So whole world is started working towards this definition to make a single sensor. Otherwise, one can say, okay, I will take and extract it and then do the analysis, everything. That also can be called as biosensor. The biosensor means is a single unit, a device. So this IUPAC definition says that it should be a self-contained integrated device. It is not parts. So if you do a part work, it is considered as only analysis. It is not considered as a biosensor. So this is a multidisciplinary expertise required for the biosensor. It is not like a physics or chemistry, etc., where you have got a syllabus in which you are going to work. Here it is necessary where you got expertise which are related. Here you have to work on a biosensor on unrelated aspects, totally unrelated. For example, one has to work on the chemistry. Chemistry people got the synthetic recombinant molecules. The biology has got the natural recognition again. You got the molecules. The transducers are mostly with physics people, electronics, etc. So all these people has to combine. And when so many people combine with different disciplines, there will be some misunderstanding initially. With the time only it is possible for them to understand the concerns of the other. And there is an interface is there. One interface is there. Here, how to put this sort of a chemical here on this one, it will be the big subject. So, how to put the recognition molecule on transducer itself is an important aspect, which is also depends upon the all sciences. It is a physics, chemistry, the physics, chemistry, everything will be involved there, because the shelf life of a biosensor will be depending upon how we are actually able to put this a biological molecule on a transducer, which the transducer may be containing a gold or a platinum, or it may be containing a carbon, or it may be containing silicon dioxide, etc. So that is a technology which requires the 2-3 disciplines. So and that is going to determine the life of a biosensor. Who is want a biosensor which is going to work for 1 week? We need a biosensor which works for several days, and we will be able to put it in the fridge, and then we will be able to use it as and when we want it. Currently the glucose chips are working for about 2 years. It was not the case much earlier. So you need that whenever you get a kit, anything, it should have a long shelf life. Lot of people are able to develop the kits for the detection of some of the bacteria viruses. But the problem is most of the kits will die within 15 days or 1 month. So shelf is important. In the biosensor also shelf is very important. That interface is going to make a large difference, even though it is not much emphasized. What are the components in what we can use in biosensors? One can use the carbon nanotubes and the nanomaterials like gold or platinum, palladium, so many things, practically anything. Particularly I am talking from the electrochemistry point of view. And transducers have given something, but I have increased the number of the transducers that are can be utilized here. And technologies that are used is practically every technology. You can use the technologies in biology, the technologies in the physics like photolithography and silicon technology, etc. And technologies in chemistry, etc., where the preparation of molecules and impeding molecules all can use it. So disciplines are there are so many disciplines. So everybody can contribute in this one. No, what is the application? Applications I have told you about military applications where we are interested in finding out some of the biological warfare agents. Actually, the applications are immense. For example, if you want to do clinical diagnosis, is one of them only, where you want to find out about typhoid, malaria, etc. All these things it is possible to find out, or you want to find uric acid or etc. It is possible to find out. Farm, garden, veterinary. These farms and veterinary etc. They also will be suffering from these diseases because of pathogens. That is useful. And fermentation control, microbiology, industrial effluent control, all these things. For example, somebody wants to find out the presence of a pesticide in a polluting the water, everything. One has to use it to extract it and then give a cost instrument called GC. But suppose you got a biosensor which costs you only 20-30 rupees, it is going to be very cheap and then very fast, and one will be able to analyze it. So the biosensor has got application in several fields, etc. And now it is new type of biosensor coming where at the point of care diagnostics, where you can take the device to the patient and then you will be able to find it. Now I will come to the actual science of the biosensors. There are two types of biosensors there. One is called the catalytic biosensors and other is called affinity biosensors. So these are all the things is that the catalytic biosensor is the kinetic devices that measure steady state concentration of a transducer detectable species. There are so many types of enzymes are there. All enzymes has got some substrates and supports everything there. So if you wanted to find out the glucose, you need the glucose oxidase. If you want to have cholesterol oxidase is there like that, or if you wanted to find out the peptides, there is an acetylcholine esterase enzyme there. So these enzyme based type of sensors which have got something to do with the kinetics are called the catalytic biosensors. I am going to give some examples. Now affinity based sensors, but supposing somebody wants to find a virus or a bacteria, it is not possible to use catalysis type of thing. One has to use a recognition molecule which are based on the antibodies everything. This is a subject of the biology, chemistry everything. For biologists it needs to be very rudimentary. For chemistry people they may think that some are rudimentary, but it is necessary to know all these things to appreciate finally the biosensor. The development of the biosensor is based on the first and second and the third generation. This is based on the receptor. The receptor is to be how to put receptor on the transducer. This is one of the important aspect. So that was gone into the first and second and the third generation. These things will be giving about the this itself has become. Now what is happening? You can have. These things will be very clear when I give the examples, and right now please accept that the developments in the biosensor is based on the first, second, third generation, depending upon how the recognition element was kept on the transducer. And one has to do immobilize this thing. This comes again on the barrel.
Can I ask you one question?
Sir.
Sir, you have shown that first, second, and third generation of biosensors.
No.
In all the generations, we have shown transducer.
Yes, sir.
So you said that transducer is compulsory for the biosensor.
Yes, minimum requirement.
But there is biosensor known where there is no transducer. Where people can use some other techniques like fluorescence, some other things like FIT. People can use fluorescence energy transfer. People can use.
Agreed. Actually, what is happening is fluorescence also you need an instrument to find the fluorescence because some chemicals will be able to fluoresce in the and then fluorescence can be seen by naked eye sometimes. Or we need a instrument to do it.
Not by the naked eye instrument, but if you use the FIT analysis, then we don't.
FIT analysis, yes.
Then we don't require any transducer. So where we will classify those biosensors?
Agreed. You are right. These classifications has come before they are using the again what we call the FIT has come into the picture at that time. So the definitions are not modified. It is normally considered you need a transducer for that one. The FIT is what sensors and that will not fall into this one, and there is no special definition is not given for that one to the best of my knowledge.
So this definition this classification should be modified.
Because I have seen lot of papers because lot of new technologies are coming, so it is fitting them all of them into one thing definition available from the IUPAC is difficult. IUPAC has given one definition which I showed you, International Union of Applied Food Research. These things what happen, the fluorescence, the compounds which will be able to fluoresce in some on-off conditions, they are not included in this one. It is only for definition, but they are also considered to be the biosensors. So this is what because what is happening, the technology is changing again widely. What we are using today, the names are also not becoming, which I am developing the subject. Definitely these sort of questions will come, will be able to answer. Now these are immobilization methods are also important when we are talking about this sort of biosensors. So biosensor, when it started actually, it all started with the measurement instruments which will be able to measure the oxygen and also hydrogen peroxide or pH conditions, everything. So now they are becoming the smaller because of the microelectronics and other type of these technologies. The basic things will remain the same. Only thing is the improvements are there in the way we are developing. So first they depend only on the pH condition of oxygen. Now no more that but they are dependent. These things are changing, which I will be telling you again. So these are simple tools we have made in our laboratory, and then recently people are working on these photolithography etc. by using the amperometric, potentiometric etc. These implemented about devices they are doing it. Second thing is that all types of sensors cannot be made by using one type of transducer. So it needs to be depending upon the necessity we have to use the a particular transducer. I am basically electrochemist, and then the electrochemical devices, the publications are increasing in this one. These are the following reasons why we need the electrochemical devices. Basic electrochemical sensor detectors are suitable for on-site detection because they are very small. They doesn't require any optical instruments, everything where you will be doing it. Then high sensitivity selectivity wide linear range is possible with electrochemical devices, and then they got inherent miniaturization of such devices possible because these electrodes can be made of the millimeter size, or it can be made micron size and also nano size depending upon technology we have got it. So it is possible to miniaturize. Why you have to miniaturize? Nobody wants to have a big sensor. Sensor means the concept is that you must have small device which will be able to detect so many. So if you want to detect the multiple type of analytes, it is possible to do by putting so many electrodes on a single platform and then do it. Second thing. So ultra microelectrodes, everything will be useful. Then compatibility is with nanotechnology is there with for the electrochemical devices, which I will be telling. Now the wide variety of materials can be used. So as I told you, it is based on the transducers. Then wide variety of materials can be can be utilized. And if you use an optical device, mostly gold or something can be used, or if you use the electrical devices, mostly silicon dioxide or silicon or zinc oxide will be used. But whereas electrochemist offers several types of the surfaces. The surface become very important when you wanted to put this this sort of a particular to immobilize the bacteria etc. So the types of possible in situ polarization entrapment is additionally possible here. Some of the one method of keeping these enzymes or the antibodies on the substrate is to entrap them in some matrix, and some of them are called continuum polymers. In this particular method, it is possible to develop by using the continuum polymers. And continuum polymers can be prepared in situ by using electrochemically. If we add the monomer and then this the biological material, it is possible to just to prepare these things and immobilize them in situ and for a biosensing. Now I will try to an example about very well known is the glucose sensor, how it happens, and biologists know much more better. And actually what happens if you wanted to detect the glucose, the best thing that anybody biologist will tell is that you take the glucose oxidase because it will be able to recognize. The biology part is something different. What happens is that finally to a sensor man is that there is a FAD portion is there in the whole the glucose oxidase, which is responsible for the oxidation reactions of the reaction that is taking place in the enzyme. For example, if you take the glucose will become glucolactone etc. by this small moiety which is embedded in a sheath. This is going to be the how we will be able to get information is going to be but tell us how we are able to make the biosensor. Now this can be given in this picture. Supposing you take glucose oxidase that is shown in a green color, and if you put that glucose, it forms the glucolactone or the gluconic acid. So from the biosensor point of view, one can find out the changes in the gluconic acid because the pH changes are there, and people initially who worked they have based on the pH transducers, and then but the problem is that the concentration range will be the changes that are going to have pH will be very less, and that's why it is not a good device for the practical purpose. The other thing that what happens is when glucose is getting gluconic acid, the other is oxygen is going to become the hydrogen peroxide, or we can use artificial things like a chemical called ferrocyanide, ferrocin etc., which will do the job of the oxygen. Oxygen actually will take electrons and then become the hydrogen peroxide. So same thing will be done by the oxygen mediator. Now the whatever you are getting in the shops, it contains a ferrocin molecule which will be able to give you this sort of reaction, and then the electrons from the oxygen mediator will be going. There is a lot of theories there. If anybody interested of lecture, I will be able to tell about it. So compared to oxygen, if you are using the concentration range in which you can measure the glucose will be depending upon the concentration, which is all dependent upon temperature, and then also efficiency of the electron transfer between oxygen to the glucose oxidase. Whereas if you use a mediator, it has got much better efficiency, it will be able to give you the good electron transfer, and then also it will be able to give the a better linear range than the oxygen. So this is what is practically what you are getting in the market is the one based on this principle. Supposing somebody wants to find out the acetylcholine, suppose somebody wants pesticides, one can use the enzyme on the principle of the inhibition of the enzyme called acetylcholine esterase. Our laboratory also doing what a lot of work on this one. These are taken from literature. This enzyme will get inhibited if you contaminate with any pesticides, etc. So what is the basically to do is the acetylcholine esterase will be able to hydrolyze this one, this acetyl compound to give the acetic acid and the thiocholine, two compound will give. So it is the basic reaction it will go. If you convert into electrochemistry, this thiocholine part can be oxidized by electrochemically, and now a lot of people electrochemistry started with the platinum electrode, carbon electrodes, etc. to oxidize that one to disulfide, and then to yield a current which can be which represents indirectly the activity of the acetylcholine esterase. So this is how it is what is positive activity. Now supposing this enzyme comes in contact with a pesticide, activity will come down and the production of thiocholine will also will fall down. So that can be founded by using the cyclophotometry experimentation. So this is how this sort of a thing it is possible to find out, and these are called the catalytic biosensors. We are mainly working on the immunosensors, which I will be talking about it. So the biosensors are two types. One is the catalytic biosensors, which mainly depending upon the enzymatic action. Other is all the affinity. So affinity based biosensors are there by using the antigen antibody based biosensors. It is also called immunosensors, and the other is called DNA blood based biosensors. So we know about DNA, and with the help of DNA also it is possible to find out. In the DNA, what happened is that they will take this DNA of a sample and break it by using their chemical, their biological methods, and then make a representative called as a probe they use it, and then they will put another sequence from the patient sample, they take it, and then they will put two together, again it will hybridize and form a DNA. Then what is the difference is that every the change in the structure of DNA is going to have an effect on the the sensing. Basically what happens, two strands will come together to make DNA. This is only happening in all these things. This is only will change the sequence of DNA, all these things will take ACDG etc. will change. So that particular sequence will be discussed with the biologists, and then we are preparing that type of sequence depending upon what type of disease everything, and then synthetically it is possible to prepare. Once this happens, when you form the stable DNA, it is possible to intercalate certain compounds like donomics and everything, which will be able to give a electrochemical signal. So otherwise what happens, these probes can be having some amount of, you can put a ferrocene signal, or you can put a color signal etc. You will be able to fix it, thereby is also to do this sort of a work of the by using DNA. We will go more into the immunosensor, which we did learn about. Now I want to tell those who know about antigen antibodies, it is nothing, but those who are not aware of this antigen, I want to tell. What actually happens is that supposing somebody got affected by using any bacteria or virus, everything, our immune system will produce something called what we call antibodies. These antigen antibodies can be represented as Y. A good complex forms between the antibody to the antigen. Supposing we by mistake we take a food or anything or inhale a bacteria or something like that, then what happens, our immune system is containing some antibodies already in our body, which will be able to do. And this this comes completely under microbiology, and then these two will form a complex. Now this what happens only. So this is the only way of finding out the presence of a bacteria etc. in a given sample. So biologist has done called ELISA, which I will be telling again. So what is happening, by the antigen antibody you get a complex. What changes, the weight of the material will change. So a biosensor man can think whether he will be able to use a transducer based on the gravimetric principle, and there will be some charges, some charge distribution, everything will take place, because these two molecules will come together. They are possessing initially some charges. The charges may come down, are changed or get nullified, when you can use by conductive methods, whether you will find charges, this is one thing. So like this, what happens, one has to see what type of transducer has got it and how he will be able to do it. Now what biologist is done is that it is not possible to see, because these are all on the nano dimensions and then one cannot see all these things. So what happened, they have used on the left side, if you see antigen antibody complex revealed by antibody conjugate enzyme. This Y is the one which is put in a platform, that is one antibody that is possible to do, biologists do it, and then the one which is like a star is the antigen that is taken from a sample. So these are formally samples, just take a showed on the right side, and then to know this happened, which is not possible by any means initially. So what they did is that they took another antibody which contains a tag. At that time they used enzyme. It is possible to use enzyme as a tag. Enzyme has got specific reactions, and then they multiply also. They got very good the formation ability of the products, and then by the reaction of the enzyme they say that okay, this whole thing has formed, and then they will be able to find the presence of a disease causing the antigen or a bacteria in a sample. This is what is done. So here normally in laboratory practices, to use either HRP or use the alkaline phosphatase, they will be using it. Now the labeling, how to use it here. Previously they used the enzyme. They use some people use radioactive labels, but that is not good. So now the practice has reduced. Still people are working on radioactive immunoassay, and then quantum dots, metal complex, whichever can give you some signal, can be attached to the antibody, and thereby we will be able to get a signal. This is the basic principle of the sort of immunosensing. Now this is an enzyme linked immunosorbent assay. Just I want to tell what biologists will do. So we use this similar principle to just to find out, and we will get it. They will have a plate on which they will be able to put this antigen or antibody. There are different formats are available, and then they will put in. So this practically whatever I showed you earlier, but these things can be revealed with the help of a plate on which they put this sort of a solution containing the antibodies, everything that will form a but then be adsorbed on the plates, and these plates are available commercially, and then they put secondary antibodies, all these things, etc. Finally they have something which will be able to reveal this whole formation with the help of enzyme. So many formats are there, depending on format they use it depending upon the type of disease, whether they want to find antigen or antibody. These kits are available in the market also. Only thing is these are a bit costly. The sensitivity level is slow, so that's why biosensor people are trying to replace these kits. Now what we are doing is we are trying to do the similar thing in an electrochemical fashion from the electrochemistry field. So what basically what you have got a tag. The tag will be able to give you some signal. So the whole while following the principles of biology, it is necessary for the electrochemist to know how to know the presence of a tag, and then its and also understand the signal that is going to come out. In the case of the biologists, they depend upon the optical signals, which are not efficient and highly sensitive. Electrochemical signals are supposed to be sensitive, so we are working on the electrochemistry aspect. So what happens is that but you must have an enzyme which will be that enzyme. What we can use, we have taken the alkaline phosphatase as enzyme. This enzyme was already tagged to the antibody, etc. What will happen to this one is that when the alkaline phosphatase enzyme was kept along with a compound of substrate with the para-aminophenol phosphate, we have given an example. It will form the para-aminophenol. So the characteristics of the electrochemical parameters are understood by on the first PAP. If you can see the PAP is this one. Similarly, if you have got this is PAP, and if you got a phenol instead of that one, you can get this sort of a compound phenol also. It has got this sort of a chemical, this sort of electrochemical properties. But suppose you use one octal and one NP, you have got a different characteristics. Now the question comes, the selectivity the portions, and then the basal concentrations and the enzyme kinetics. All these things in the concentration. Finally, we have did lot of work and then found that one octal the phosphate is able to give reproducible results, and which is devoid of any the base and everything. So we are able to do it. Now how to do this one? for this one we need an electrode which is efficient enough to give the electrode. That's how the biosensor concept starts there. So we need an electrode which is efficient to give. We are giving the different types of the electrodes here, both commercial and also the one which are prepared by us, and then there is a difference is their efficiency. This efficiency we found that some of the electrodes are very efficient, and then we have gone for the development of the biosensor. We were just exploring the different possibilities of technique, and one is called the QCM, the quartz crystal macrobalance. The physics people use the quartz crystal macrobalance as a for finding out the deposit of the gold or aluminum or platinum that they are doing on a substrate. So if they wanted to know how much is there, then what they do, they will keep this instrument in the vacuum deposition unit. The water is deposited will be deposited on the crystal also. A crystal has got a special property. Its frequency is going to be depending upon its the weight on the, available on the crystal. So it is nothing to do with biology or biosensor, nothing. But such as it is a transducer. Such a transducer they are trying to convert into a biosensor. So these cells are available. So we have got the transducer directly. We didn't make anything, but they are very costly. They are about 3,000 rupees for each piece. So what is the advantage that previously we are using an enzyme as a tag. Here we don't need any enzyme. What we did is we have taken the crystal and then we put in the mercury stickers everything, and by self-assembled molecules we are able to fix the COOH group onto this one. And then by using the EDC and NHS chemistry, we are able to fix the antibody onto the system. So what happens, you are not using any enzyme tag everything which is maybe difficult, or stability everything will come to picture. Without using them, we are able to find out by QCM sensors we are able to find 3 cells of the bacteria which is more sensitive than the electrochemistry also. It is more sensitive. Only thing is very costly, and then the specificity becomes very important on the sensors which doesn't use an enzyme, because it is this we have done it, but we felt it is going to be very costly technique, so we didn't do much. But we applied this technique to 2-3, and then they found that generally the sensitivity of this instrument is very high, but it is not portable. So we are trying various options and then trying to find out. Now this is the detection of the malaria falciparum. What is the idea is that to detect the samples of the malaria. Here idea is that these are the electrodes have already we got same electrodes. The electrodes has got some amount of efficiency to oxidate the compounds and give some signal, but we want a signal enhancement. Everything else is same. We want to have only signal enhancement. What we did is that we have modified the electrode by using the carbon nanotubes which you can see in the CM, and also by putting the gold nanoparticles by using the electrochemically. So we first modified our electrode, and then we have put the same antigen antibody, that is same story continues. Now in order to know whether we are able to have a better sensitivity for a given without any antigen antibodies everything, we are able to do a lot of electrochemistry, and with unmodified electrodes what we got that one if we showed earlier, it gives the green current which is very low current. But supposing you modify the CNT and other things, because of their nanomaterials they will be able to give more amount of current, and we are able to get very high sensitivity for our particular analyte. Now what is happening, if you see that we are able to film this the malarial falciparum, what happens, what I was told that is when the malaria gets into the person, it secretes something called that PHRP. It is a material that was chemical that was secreted by it into the blood. If you find out the presence of the PHRP, then you will be able to find out what that the person must be having the malaria. This is indirect indication of that. It is called PHRP2, PHRP3 are available. We are able to get PHRP from all the biologists. Now what is happening is that we want to keep practical use, and this immunosensor part should come this side actually. It should not be there here, it should be there and then paracheck. So what I did is what our colleague showed is that there is something called paracheck is available in the market, which will be able to find out the sensitivity whether a person has got malaria or not. That is from Guava it comes that kit. So they have got some parameters called specificity, sensitivity, etc. Some calculations are there. We have gone through calculations, and we can see that this is for the paracheck. the sensitivity is 75 and the specificity is 81, whereas for immunosensitivity is 96 and 94. How we are able to get? We went to the Malaria Research Institute here, here itself, and then our people went to Bengal and other places also where the malaria somewhere is prevalent. It is the intensive work on the from biologists. They are able to get samples. Now we are able to get these sort of different. We are able to get nearly about 103 samples or so. We got it. So here also we can see that we have taken different samples. Now the problem is that the fever doesn't come because of malaria. Some people are suspected to have malaria. They don't have malaria. They got the fever because of some other infection, something they got it. So it is necessary to know. So we have pulled lot of samples who got the disease, who don't have disease, who and were suspected to have disease, and also natural. So this area which you are taking, this is an ampermetric curve which gives me the current. You can see that here majority of the people are having in this region, they are able to do this current. And we have again calculated 160 plus 3 into 20 is equal to 220 nanoampere. Okay, that is the current you are getting for a normal human being. Same current is obtained by people called who are supposed to have the who got fever, but not because of the malaria. It means they don't have that falciparum in there. So this is how we are able to calculate. Anything above, from here you can see the person has the highest disease, I think, they too much have, and then these people are anything above is considered to be the positive. So what is happening? We made a background based on the patients and the non-patients who are the patients who don't have malaria and but who got fever, and those people who are healthy. All these things we have done, and all these are followed in this region. This is or this become more background, and people who are having higher than this one are considered. This one, etc. They considered. That's how the calculation of the specific all these things done were able to make for the again for the paracheck compared to paracheck. Our immunosensor is much higher sensitive. What was found to be higher sensitive is a 103. But if you want to put it into the market or something, you have to improve a lot of things which I will be telling you later. So this is another thing that what we do is by using the antibodies. There are two methods of finding out the disease, either by the antigen, that is the best if you can find out. Otherwise, if antibodies are produced by this one, supposing a patient goes to a doctor, at some stage when he knows he will go, initially the antigen will be increasing and later then the antibodies will be increasing. So the test is to be done accordingly. That's what we are told. So we are trying to put, and always we try to compare our results with the standard biological sample testing. Now what is happening? Our interest is basically being electrochemistry, which material is going to give me more sensitivity. Resin is a toxin, and then it is based on and castor oil. I don't know anything. I think nobody would have taken castor oil in this young generation. But in my generation, that is where when I was very small kid, they used to induce the density everything by giving the castor oil, and some children used to die also in those days. It is all because the castor oil that what is used for the outside will contain it contains a toxin called resin. This resin is being used to kill people. One Bulgarian person was killed by the some countries to just it is used as a biological warfare agent. Some people call it as chemical warfare agent, but he was killed and it was very well known. And there is if water etc. is contaminated, there is a possibility that the people may die. But because this one, so it is necessary to find out the presence of the these sort of things in the water, particularly in war times, etc. Because we do not know any terrorist, everything may be using this sort of the material. Maybe terrorists or over enemy country, or it may be sometimes the terrorists may use it. So what is my interest is to how as I am getting the benefit out of the nanomaterials in getting higher sensitivity. At the time the nanomaterials started coming around, that time they are talking more about it. So we want to know practically whether I am going to get a sensitivity for these things. So what I did is that we did a lot of electrochemical characterization. If you are interested, anybody interested, I will be able to tell about that one. This tells you the best conditions for mixing a particular mineral oil along with the with this one to make a just a paste. If you take SEM, the SEM paste looks like this, etc. But normally the carbon paste filters are very well known in electrochemistry, and they are being used mostly by using the graphite, etc. for the analysis. And then the cheapest things that can be done by any university or anywhere, it is very easy to do this job. We have done the electrochemistry of this one, and then we have done the detection by using the carbon paste, etc. The sensitivity of a biosensor is going to depend upon n number of factors. One is that what is the efficiency of the electrode system to give a current, what is the amount of the adsorbers on the material, that is our antibodies are able to absorb. These two factors will make a profound influence. It is not possible to resolve these two factors. How much is sensitivity part, how much is absorption part, that is very very difficult to make out. That I will be telling you later. But what finally we found is that the MWCNT, which is having the carbon tube, is able to give me 4 times more sensitivity under the same circumstances. Means we are not using same antigen antibody system is used and the same methods are used. Finally, my result is that I am getting a better sensitivity of the order of 4 times better by using MWCNT compared to this one. Now, what is the implication of this higher current is that supposing you go to an electrons, finally you have to make an instrument, you have to go to an electrons person. The electron people are very happy to develop an electron circuitry for microamperes, etc. than nanoampere and picoampere. It is not that they are not capable, they are capable of doing it. Only thing is nanoampere currents, etc. they pick up the signal even from the surroundings and then give a lot of noise. So if your sensor is able to basically give very high current, you can make a cheaper instrument. So this sort of a data will be utilized, will be useful. One should keep in mind to get a higher current, so that will be useful. So this is how we are able to get high sensitivity. Because of this one, we are asked to write some review articles also. This paper has got good name at that time, and then we are also do.
Excuse me, sir.
Yes.
Sir.
Yeah.
Carbon nanotubes are said to be giving a field emission current density of 10⁻10 per milliamperes per centimeter squares, right? And you're measuring current in nanoamperes. Was there a deliberate attempt from your side to measure it in nanoamperes?
No, field emission is totally different issue. It is nothing to do with electrochemical current. In electrochemistry, the current comes because of the oxidation or reduction of the compound.
Right.
You are talking field emission, etc. That is totally, that is you are telling disparate devices you are talking about it.
Yeah, we apply the field and the current.
No, this is nothing to do. Here the CNT used it to as a material for electrochemistry, like a platinum or gold or carbon you are using it, they are used. The current is a function of the concentration of the compound, surface charge electrode, and the efficiency of the electron transfer between the compound to the CNT. It is nothing to field emission, is totally different issue. You are talking about discrete devices and then you apply voltage and then that is not totally different issue. This is different issue. Means there is no comparison between these two things in MLSR. So that was used. I think SSPL is working on this one in Delhi. SSPL is working and in Bangalore one more laboratory they are working. We got a common project on the gas sensors. At the time they used to talk about the field emission and other things. Devices. So this is how we were able to get. Now then for SCP we have done, already we did it. So far we are using enzyme labels, other labels, everything. But whenever we put labels, etc., you would have seen that you have to add a substrate, or we have to do something or a substrate like for naphthyl phosphate you have to add for first thing. Otherwise quantum dots you have to keep in the HCl, extra time is taking and then they get dilute everything. So we thought that why don't we have a direct transduction between the antibody to this one. So this is the latest work that we have done it. What happened, this was I will publish this one. What we did is that we have taken the ferrocene as a probe. The ferrocene has got a very good electrochemical properties. And ferrocene carboxylic acid was conjugated to the antibody just like we did by EDC and its chemistry, and we did it. So they are able to call this is called ferrocene molecules to be considered. And then gold nanoparticles were used to enhance the electrical activity of electrode. The problem is that the distance between the ferrocene to the electrode should not be very high. So what we did, we have put the nanoparticles, etc., so that some environment of electrical environment will be there around that one. So we are able to do it, and we will cyclophotometry and all these things we are using. Finally, what is happening? I am not using any enzyme. We are not using any substrate. No additional chemicals are required. No dependence on external vendors for conjugation everything, because mostly you depend on external if you want to get the conjugates from the outside. So all these things are removed from here, and here you can see how the electrochemistry of this simple electrode after the modification is improving for just the ferrocene. So this is how it is improving. So now what is happening? The direct electron transfer is taking place. So you are not getting any chemical. Once you put the antigen antibody interaction over, you can straight away get a signal from the scalar photometry. We are able to get the sensitivity directly. such a technique, if it works very well, it will be in future it will be able to be useful as a biosensor. So now what is happening? We wanted to know, at the time he was using, now he is working in Parley as assistant professor, and at the time was JRF. And then what is happening here also again, we use the two materials for the good electrochemical communication. One is the carbon nanotubes and other is the graphene. Again in the graphene, the sensitivity levels are not too much high between these two, but the current is very high in the graphene. Again, if you want to make an instrument, it is going to be cheaper for this sort of a work. So, in conclusion, just this conclusion, successful biosensor development depends upon the ease of use, cheap portability, minimum number of steps, everything that are required. Then long shelf life is required. People may say a lot of things, but long shelf life is the one which is probably even the techniques. Repeatability and reproducibility are very important. Then suitability for mass production, even though very good sensor we make it with lot of components, it's not going to work. You need a technology which is mass production. Relevance to the measurement. Suppose glucose is found at 100 to 120 milligrams per deciliter, etc. Whereas the antibodies are found at a very nanogram level. So same technology cannot be used accurately for these all the things. So this is one thing. And electron biosensor has all the qualities at the as proved by the glucose sensor, and I hope it will be useful for any disease diagnosis also. Thank you very much.